Anybody, any errors? Any doubts?
Hello, I've got the BCF file.
Okay, that's great then. What about ye, sir? I think you have joined after lot like after break. Okay, not an issue. So we have run the complete pipeline for the DNSA, and now we are at the last step of it, right? Like we have got the results also. Now we have to understand what is actually present in the VCF file and how that it is storing the variation information, because as we are calling, DNSSEC is used for the variation calling only. So how we can know about that variations, that we are going to see right now. Okay, so you can try to open that VCF file into the Excel. Let's see what is actually present inside that. Okay, I'll also share my screen now. So just a moment, give me 1 minute. I'll take some water. So here you can see we are having this VCF file by the name of GATK output. So let's try to open it in Excel format now. How many of you have seen this VCF file previously? Um, Okay, so you can see, I hope this VCF file content is properly visible to you all. So can you please tell me what is actually this VCF file? If you haven't seen this, at least if you're knowing any description about it. Okay, not an issue. So basically this VCF file format, it's standing for the variant call format. So it is actually the standardized file format which is commonly used to store the genetic variation information which we have obtained from the DNA sequencing, DNA-seq data analysis. Or maybe if you are processing from the NGS, so this VCF file will represent the variation information specifically. Okay, so basic structure for the way this VCF file we are having as a like it's a tab limited. Obviously here also you can see these values are tab limited, where each line is going to represent certain genetic variation which is found in our genome or which is found in our DNA. So first you're going to have some header lines. If you will see, This VCF file will basically start with this header section, which is nothing but the metadata, or you can say the information about the data which is present within the file. Okay, it is the header information, and metadata is nothing but the extra information about your headers. Okay, this will include which is the reference genome used, or maybe it will be having some extra information. And then below, if I will scroll down from this 26th line, you can see we are having the actual variant records from the 27th line. Here, first of all, you are having the metadata, you are having the header line then, and then from here you are having the variant records. So, like basically each variant record will represent some genetic variation which is identified in our data, and basically these variations may include the SNPs or insertions or deletions or the structural variants also. It's totally depends on the algorithm which you have used previously in command by the use of GATK. Now here, if you will see for each and every header, let me just highlight this header so that it will be more properly visible to you. So now if you will see, these are the headers for your main information. So how you will be able to know about this headers, right? Because in the info, if you will see, you are having some information like AC, AF, AN. So what is this information is actually standing for? How you are going to get that information, right? So that information you can just read it here in this metadata. So here the description for your each and every header is being provided. Okay. So first you can see you are having the filter. Here you can see filter column is there. So filter is what? If you're having the low quality Fred quality score, so that information will, if you're applying the filter, or if the tool is taking that filter, so it will just provide that information. In the format, if you will see, you are having AD, DP, GQ, GT, PL. If I will show you here, you can see GT, AD, DP, GQ, PL. So what is this terms are standing for? What are the short forms are for? So that information also you can see it here. Okay, allelic depth, read depth, genotype quality, genotype. Likewise, you are having the information. Now let's talk about this columns first of all. So basically it is having various columns over here, so which is giving us the information about our variation. So first column we are having as a and you can see sources also being provided as a haplotype color. With the help of that only we just have found out this variations. So first column we are having as a chrome column, or you can see the chromosome, right? So here you are going to get the chromosome name or the identifier where our variation is located. That location you are going to get, chromosomal location. Then here POS is nothing but the position of the variant on that particular chromosome, and these are the coordinates basically. As of now here, if you are having the SNPs, means only single position will be provided. Then you can see you are having the ID column, so it's actually the unique identifier for our variation. Then you are having ref, means it's a reference allele at that variant position. Means on your reference genome, this particular base has to be present, but it got mutated and it got altered. Altered means what? Alternate allele which is observed at that variation position, at this particular position on chromosome number 17. So T got mutated into C likewise. Then you are having the qual, which is nothing but the standing for a quality. So it's a quality score which is representing the confidence of the variant call. Again, if you are having the quality score more, means more confidence is there likewise. Then here we are having the filter. So basically this filter status will indicate whether the variant has passed certain quality filters or not. So basically this information about the quality of variant call, or whether it has passed certain quality control filters or not. Suppose if you are in the command only, or in the GATK tool, or maybe while performing the pipeline only, if you're applying some kind of quality filters, So if that tool is taking that filters or not, or if any by default filters are being mentioned in the tool by like in its algorithm only, so that filters, if these filters are passed by your variation, then they will just mention it out over here. Okay, so they'll just mention it like pass, or maybe we'll be having certain another information. So you can see, suppose here they have written it as a pass. Means it will indicate the variant has passed all quality filters over there, which are applied during our variant calling, and it will be labeled as a pass. But as of now, we haven't applied any filter, so that's why it is representing it as a dot. And by default, filter is also not present for our tool, like this. Then further, we are having the info. So basically, this info column describes the format of the genotypic data actually for our each of the sample which is included in our VCF file. So it will specify the structure of genotype information, like including genotype calls, what is the read depth, what is the genotype quality, and so many fields will be provided. So let's talk about each and every field in detail now. So now you are having a ACAFAN. So let's move bit up, and here you can see you're having GT. GT is nothing but the genotype, means this particular field will represent the genotype of the sample at the variant location actually. So basically, like genotype are typically represented using the combination of different digits. Okay, like which are separated by some slashes or maybe some pipes. So if you're having, maybe I'll write it down over here somewhere. Here, if you will see genotype, so that is represented here also. So if you are having the genotype in terms of this, like whatever the format they are giving you here, na, this GT, AD, DP. Okay, I was talking about this ACAF and I got like shifted in between the GT. Okay. AC is what, if you will, not AC, we are not having that information over here. Info, yes. Here you can see it's AC. So it is nothing but the allele count in genotype. Whatever the altered alleles are there, that information you are going to have. AF is nothing but the allele frequency. Then AN is nothing but the total number of alleles in a called genotype. So here you are having that information. How many allele count is there? What is allele frequency? What is that number? So you can see DP is also there, which is nothing but the depth. So for each and every field, because a lot of fields are there, you can just read this description about each and every term here. Okay, you are having MLEAC, which is maximum likelihood expectation of the allele counts. Different algorithms or the methods have been used to do that. Then here comes the most important column, which is what format we are having. So in the format, you are having GT, which you can see it here. So in the respective column, in the genotype, in the allelic depth, in this DP, what are the counts for your variation, that you are going to get it in the next column. Here you, if you will see, sample ID is being provided. So GT is 1/1. AD is 0, 2. Likewise, the information is provided over here about the terms. Okay. So now, as I was talking about the genotype, which is going to give you the actual variant locus, like what is the genotype of that sample. So now, if you are having it as displayed to be 0/0, means it is, this particular variation is homozygous for the reference allele. Okay, if it is showing you as a 0/1, means what? It's a heterozygous. Like with you will be having one copy of the reference allele and one copy of the alternate allele. Likewise, you are going to have homozygous, heterozygous. And then if you're having 1/1, if you will see most of the cases in our sample, we are having 1/1 only. Here you can see you are having a 0/1, means it is representing the heterozygous allele over there, right? With one copy of reference allele and alternate alleles. Now here, okay, 1/1 is taken as, okay, first Jan. So here if you're having 1/1 means what? It's a homozygous for the alternate allele. This is what the genotype you're having. Homozygous for alternate allele. Likewise. So this is how this genotype will be represented into your sample. So yes, if you just want to filter out the heterozygous alleles, then you can just filter out with the help of this genotype, GT it is. Okay, that description you can just find it out here also. There is GT. You can see. Now let's talk about the AD. AD is nothing but the depth. So basically this DP or the depth is going to indicate the depth of coverage for variant locus in our sample, and it will represent the total number of sequencing reads that align to that particular position, and that numbers also you can see further. Then we are going to have the uh, AD. Okay, here's I have explained you the DP is nothing but the depth. Then we are having the AD. AD is nothing but the allelic depth. DP is nothing but the total depth. AD is nothing but the allelic depth. Okay. So AD will provide you the breakdown of the total depth into the number of reads which are supporting each of your allele, like reference and alternate also at the variant locus. Like if you are having the AD as 10,5, okay, it will indicate that there are 10 reads which are supporting the reference allele and 5 reads are supporting the alternate allele. Likewise, you are going to have the information. Okay, here 0,2 is there, means what? It will indicate that reads are not supporting the reference further reference allele, but your 2 reads are supporting the alternate allele. Likewise, this is how it can be interpreted. Then we are having GQ. Okay, here if you will see allelic depth, okay, I'm just explaining it again. Allelic depth is nothing but like it will provide you the breakdown of how actually your total depth is there in the total number of reads which are supporting the alleles, and at the variation location also. Like if you're having this, suppose here if you will see the numbers are being provided, right? 0,2, 0,2 means what? Like instead of this 0,2, if you're having the number like a 5,2. So what this 5,2 will indicate that there are 5 reads which are supporting the reference allele and 2 reads are supporting the alternate allele. Total number of reads are what? Here if you will see DP in the third like slash you will be having the DP, right? So here you are having the 2 reads, isn't it? So that 2 reads are supporting your reference allele, sorry, alternate allele. That is what I'm saying. Okay, if you're having here maximum depth, like if you are having the 10x depth, so out of that 10 reads, how many reads are supporting your alternate allele and how many reads are supporting your reference allele, that information you will have at that particular location. How many reads are supporting for a reference and alternate allele. Is that clear? Yeah, if you remember, I have in the introduction part, I have drawn one diagram like this. If you're having this thing, here you are having the depth. Suppose you are having 10x depth, 4, 5, 6, 7, 8, 9, 10. So on your reference genome at this particular position, some A is there, right? And this 6 reads are also having A at this location. Means what? It's a reference allele. But here you are having the alternate allele as a T. Means what you are going to have it here, the count you will be having. DP will be 10. AD will be what? Here, how many reads are supporting your reference allele? First number will be that. So reference allele is 6. 6 are supporting A and rest of the 4 are supporting your alternate. Likewise, you will be having that count. Depth breakdown will be there. Okay. I hope it's clear now. Now let's move to the next part. Yeah.
Um, Ma'am, what about the other number 69060?
Yeah, I'm
Six.
talking about that only now. I just have explained you GT, AD, DP. Now let's go for a GQ. So GQ is nothing but the genotype quality. Means it's a quality score, genotype quality score, which will estimate the likelihood that the genotype call is incorrect. Okay. It is also Fred scaled where the higher scores will indicate the higher confidence in that genotype call. So higher the scores, higher is the confidence. It's a confidence score only about the GQ. Okay. Then we are having the PL at the last 960. Okay. So what is this 960 actually? So this PL will represent the Fred scale likelihood of the different genotype calls. So these like by the statistical way only it is calculated. So lower PL scores will suggest the higher confidence in the genotype call. For example, if you will see, if you are having the PL score as 10, 20, 30, let's assume you are having this example. Here, if you will see, you are having the scores like this. I'm just giving you the another example. Now, what is this representing? So it will represent the Fred scale likelihood for homozygous reference. Okay, homozygous, heterozygous, you can interpret from here. And homozygous will be having the 10 score, heterozygous in between you will be having, and homozygous alternate genome genotypes respectively. Like it is basically representing if your homozygous, heterozygous, homozygous, this particular conditions are there, then what are the scores for that? Likewise, they are giving you the information likewise. So lesser the score means what? Better that particular thing is, or what you can see. Lower PL will suggest the higher confidence. Now, if you will see, you are having 1/1 means it's a homozygous for alternate allele. So that's why you are having a 0 score means 0 score is representing the highest confidence. For the 0/0 means for the homozygous, it is giving you the 90 score, which is two way larger, right? So this is what it is representing the confidence about your genotype. Okay. That is what you are having as a PL, Fred scale likelihood. Okay. So, and at the end, you will be having this sample column further. Like here, I have told you format. What is actually the information is there for that particular format. So, this particular, if I want to give you the description about this column, so this sample column will be having like it will be the column containing genotype information for each of your sample, which is analyzed in the format field. So that is what you are having. So basically, as I have told you, VCF file will be playing the role in storing the variation information, right? Which is we have got it from the DNA sequencing experiments. what you can do is you can just read about this VCF file also in detail in the Google also. They have also explained you, like they have also given this VCF file format in a proper way, because this VCF file format is bit uh, like complex to understand because a lot of fields are there. So you can just try to refer this PDF also. You can see here they are giving you the full information. Okay. So that's it about the VCF file format. If any doubts are there, you can ask me. And as we are just having a smaller data, we just have took the smaller data to process this pipeline. So here, how many uh, variations like we are having in that particular region is these many only. So you can just count it like till 26th, it's only headers, and from the 27 to 81, we are just having 55 variations. Maybe in your case, like might be I have interrupted this process yesterday in between because it was taking a lot of time in my system. But yes, you can just try to run it in your system. Maybe you will be having more number of variations also. Okay. So this is what this VCF file is. This is how we are calling the variations. Okay. Are there any doubts?
Ma'am,
Okay. Yes.
ma'am, can you show me how to download that adapter sequence you told from Flash QC?
Okay. Just go to the Chrome. Just type here fast QC download. Okay. And here you are having one Babraham link. And here you are having download now option. So you have to download the fast QC for a Windows. So here you can see fast Q, focal point, different links are provided. But go for this fast QC. And here you are having for the Windows or the Linux, you are having this zip file. So you have to download this particular file. I'm sharing this link. You can just try to um, paste it somewhere.
Okay, ma'am. And ma'am, sequence 1 and 2 will be of the overrepresented sequence. And the sequence 3 will be of the adapter sequence, right?
It depends totally on your data, sir. Actually, if in your data, if you're having 15 overrepresented sequence which needs to be trimmed, then 15 overrepresented sequences you have to mention it. And then you
Okay.
can mention your adapter. It's not about only adapter should be pasted at the end. It's not the thing. You can paste your adapter sequence at the topmost section also. At
Ma'am,
the end, yeah.
so the order can be anyhow, right?
Yes, it can be anyhow.
Okay.
Yeah. I think I
And
haven't shown you the bad quality data. If I will show you, see here, if our data was good, as our per base sequence quality was falling under the green colored area only. But if I will show you the bad quality data, you can see here example reports are also being provided, bad Illumina data. And here you can see in the overrepresented sequences, cross mark is there, and here you can see only one overrepresented sequence is there which you have to trim it, right? So only one sequence you are going to provide. And here the adapter content is also like not present, so no need to give the adapter content, only one sequence you will be trimming likewise. So
okay.
it depends on your data what you have to trim it. Yes, I think you were asking me something else also.
Ma'am, I was asking like, if you want to give the faster sequence, will I have to write like sequence 1 or sequence 2
Yes,
or okay, okay,
header you can give anything. At the
okay,
end, they will take your second line of your faster sequence.
okay, ma'am.
Yeah. Okay. If any doubts are still there, you can ask me. Because our pipeline is done, we are going for a post data analysis. So it should be cleared here only. Otherwise, you'll be stuck at one position and you will be not moving further for the next application. Okay, because tomorrow will be not tomorrow, a day after tomorrow will be ending the DNSA. So I hope as tomorrow will be holiday, so you will be trying out whole pipeline. And if any doubts will be there, you are going to ask me in the like day after tomorrow's session. Okay, that will be the last session for the DNSA. Okay, so revise the content, and if any doubts will be there, you can let me know. Now here, if you will see in this particular Excel file, we are just having the variation information, right? You are having a genotype information, you are having a location, coordinates, chromosomal location, but you are not having any extra information about your variation, right? If this variation is happening or occurring, then what kind of causes it is going to, like what kind of effect it is going to do, right? So that extra information or the biological impact factor, you are not having it here, right? Means what? You're not having the annotation for this particular variation in this VCF file right now. So how you can try to get the extra information about your variation? Maybe some like already some people have studied about it. So for this particular variation, you may have the information to be present previously only. Sometimes the variations you may have novel, for them you will be not having any extra information. So you can like go for your further studies about that novel variations accordingly. But here sometimes maybe these variations are already present, or extra information for this variation is being available in the databases, so that we can retrieve it, right? So if you want to get the information about the variation, we are having one tool which is variant effect predictor, which is VEP, ensemble VEP. So now for all these 55 variations, we are going to retrieve the extra information. So how you can retrieve it? So just go to the ensemble VEP tool. Ensemble VEP. It's an ensemble variant effect predictor. Okay. I'm sharing the link also in the chat box so that you can also try to open it. Okay. So now here, if you will see, this is how the ensemble variant effect predictor is there. If you want to do the annotation by any another tools, any another databases, these are also you can use according to your choice. Now, if you will see it here, only the description for your VP tool is being provided. So VP determines the effect of your variants, like if you're having SNPs, insertions, deletions, then variants, like on which genes, on which transcript, on which protein sequence, as well as if any regulatory regions are there. So that extra information it will be finding out, like it will give you that extra information. Then here simply you can just provide the input of your coordinates, input the coordinates of your variants and nucleotide changes to find out the genes, transcript in which this variation is happening and all. So you can just read about this VP also in detail. So this, if you are making this VP as like directly, if you want to download and install it in your system and you want to perform this annotation offline, like we have done it with the help of Blast. So if you want to run this VP tool offline, that information, that also you can do it. So it will be having the command line tool also, like it's basically command. You have to run the command and then you can perform the annotation likewise. So that information is also being provided here. You can just see the documentations and you can see how you can use these tools further. As of now, we are going to not make it as a, like we are not going to install it in our system. We'll be using the web interface only. So because the installation is a bit tricky and it will take a lot of time, and it's not of any need to install it right now in your systems. So we can just go for a launch VEP in a web. So let me just delete these results. I have run this VEP previously. Okay. So now here you can see this is how the page will look like. So here we are having a new job. We are having one VCF file. So we have to get that extra information about that VCF file. So you can just click on the new job, and obviously whatever the input you usually provide, according to that only you are going to get the output, isn't it? Like in first module also we have seen a different databases. So if you remember UniProt database, there also if you are selecting some fields, only that information you are going to get as an output, right? So here also, whatever the input you will select, whatever the fields you will select, according to that only you are going to get the extra information. Okay. So first of all, you have to choose your species. Maybe if you are not working on the Homo sapiens, so you can just change the species. Okay, so click on the change species, and according to that you can just say it. Like maybe if you're having another mammal or another species, if you're working on the bonobo or maybe any another organism, you just have to select, you just have to check that box and say apply. As of now we have processed on the human data only chromosome number 17, so I'll make it as a human only. Then here, if you want to provide any job title for your particular job, you can just provide it. This is optional thing. It's totally your choice. Then what you can do, if you're having less number of variations, you can just provide your variation information here directly. You can paste it out your data. If you're having a larger VCF files, then you can just choose directly. You can load your VCF file also from here. Okay, so let's try to choose that variants like VCF file from the folder. You can just copy paste the data also. Hmm. Okay, I have loaded it. Then here, which transcript database you want to use to retrieve the information, that you have to choose it. Maybe if you want to choose RefSec transcript, RefSec database, or Ensemble or GenCode, that you can choose it. I'll be going with the by default, which is Ensemble GenCode transcript. Maybe you can go with all of them. That is again your choice. Then here you are having the additional configurations also. Maybe if you want to get the identifiers. Now, which IDs you want to get as an output. So gene symbol if you want the protein IDs, if you want to get the UniProt IDs also for that variations or that transcripts, that also you can just select it from here. Okay. So whichever the fields you want in your particular output, that fields you have to choose it from here. Just go through from these fields, and whichever the information you want, that you can choose it. Okay, then additional information also, if you want to get the uh, this particular main, so if you will just hover on that main, you will be able to get the description about that particular term also. Okay, so yeah, that's it. You can just check out your fields and you just have to run it now. So this VAP will take some time to generate the results. Not some, it will take actually bit extra time for that, as we are retrieving the extra information. And now here you can see this is what the job I have run it right now for our VEP file, and it is showing me as a status queued, means it's processing for the variation. It will retrieve the information and it will give it back us. Okay, whenever it will be showing you done, you can open that results, you can download that results. Okay, so let me know if you have any doubts in this VAP. Anybody of you have run the VAP? Hello? No. Okay, Ajisha ma'am, you can try to run it. Yes, you can run it. Yeah. Okay. So you will get the results soon. So here you can see for my particular VCF file, I just have got the status as done. I have got the information now. So if you want to view the results, you can just view it from here. Okay. So say view results. And now here you will be having the graphical representation also, because I have told you like how many are intron variations or that extra information you are going to have it here. So you can see consequences, you are having it here. Intron variants are 46% out of our variants. Then 5 prime UTR variants are also there. Downstream gene variants are there. So you can just read about this extra variants also, and this is what the graphical representation is being provided. Here you are having the summary of your particular this graphical representation. How many variants have filtered out? How many variants have processed? 55 variants have been processed, and novel or existing variants. So you are having these many novel variants and these many existing variants for which you are having the extra information to be available in this particular database. Overlap genes are also there, then you can just see this extra information. And now here you are having the details, like further whatever the fields you have chosen while performing or while giving the input, that fields you can see it here. You are having the variant which you have uploaded, location, then allele you are having it here. If you will hover on that particular fields, again you will be having the description. What is the consequence for that particular variation? What is the symbol of the gene, gene ID from the ensemble, then feature type, like you are having a different fields which you have chosen it, right? So you are having the extra information about your variation. This you can see it here. Maybe if you want to download this information and you want to make this information to be available here, if you want to paste it out here for your variation, if you want to provide the extra information in this file, so what you can do is you can download this file, match your variations, and you can just paste it out your extra information. Because out of 55, we are not having all the variation information. These many variants information you are having it. Okay. So if you want to download this particular file into your system, what you have to do is maybe you can download it in text format now. So click on the download and txt. This txt file is being downloaded, which you can see it. Instead of opening it in txt, what you can do is you can open it in Excel format, and the same information which I have shown you right now in the browser, the same information they'll just display it here. Here I'm having that file, and this is what your annotation is done, your variation annotation. Is that clear? You can see this is what you are having the variant. Okay, so if you will see, first of all, you are having the uploaded variation, and this column will what give you, it will give you the identifier of that particular variation as you are providing it in the input file. It could be RSIDs also for your variation in from the SNP database, or it will be IDs which are generated by the tool also. Then you will be having the location in the second column. This column will give you the chromosomal location of the variant. Then usually in the format of chromosome number, and then you will be having the location, exact coordinate of that, which you can see it here, right? In a bed format, if you remember. Then you are going to have the allele. So it will indicate that the reference and the alternate allele for that particular variant. The reference allele is usually denoted by the REF, and the alternate allele is denoted by the ALT, alternate. Okay, so this is nothing but usually if you are having this allele, so it is alternate allele. Here they are not giving you any reference. Here you can see REF allele is being separated out, reference allele, and this is what your mutated allele you are having it. Okay, then you are going to have the consequences over here. Consequence means what? This column will give you the predicted functional consequence of that variation on particular affected gene, like whether it is gene or whether it is going to affect the transcript. So yes, common consequences you'll be having it as maybe missense variants or nonsense, frame shift or splice it. Again, if you want to focus on intron variant, so that information you're going to have it over here. Okay, so you can just see that consequences. Then we are having further impact also, how much impact it is going to cause on your particular for your particular variation. Then symbol of the gene, then gene ID, then feature type, what is actually this variation is coming from, so that feature, then feature ID also, then biotype, it is protein coding, if it is present in the exon, intron, that also you can see, and here you are having the extra information about it, right? So yes, that is what you are having the information. So that you can see it here. Here you are having the alternate and the reference allele also. Okay. So this is what you are having the VEP output. So what we have done till now is we have run the pipeline. We have got the VCF file. For this VCF file, today we just have seen the description of for this VCF file, what is this VCF file actually is, and what it is storing, like it is storing which kind of information. Then we just have retrieved the extra information about this VCF file from the VEP tool. Today I just have told you how to use the VEP, ensemble VEP. That's it. Okay. So yes, then that's it from my side for today. If you have any kind of doubts, you can ask me. Okay, if doubts are not there, then we can disconnect then. Okay, then so bye-bye. Let's meet day after tomorrow and let's end up this DNA. Again, I'm giving you the warning. If you're